Hi, welcome to Channel 11 News. Today we wanted to shed some light on a phylum that is hardly ever talked about, the hemichordates. Phylum Hemichordata belongs to the Animalia kingdom and are considered the sister taxa to phylum Echinodermata. They are bilaterally symmetrical, eucoelomate, and are divided into three body regions, the proboscis, collar, and trunk. Their nerve tissue is concentrated in the collar region of their body, and their circulatory system consists of a heart-like vesicle, vessels, and sinuses, and they respire through gills on their body. Here to talk more about the evolutionary history of a phylum hemichordate is Jamie R. Geary, joining us from her office. Please welcome Jamie R. Geary to the show. To start off, I'm going to ask her a few questions about the hemichordates. What is the diversity within the phylum like?
There are three classes of Hemichordata. The Enteronuesta, or acorn worms, are slow burrowers. They have triple body division and contain many branchial openings. The pterobranchia have one branchial opening and create a network of tubes. The stems of these tubes connect the individuals within a colony. And the last group is the Graptolithinia, which look like tiny saw blades, and they are often thought to be planktonic.
Where can you normally find hemichordates?
Hemichordata are a marine phylum, so you would be able to find them at the bottom of bodies of water, such as the White Sea and areas around Greenland and New Zealand.
What is life like for these organisms?
In terms of day-to-day life, they gain nutrients from the fine particles of organic matter through suspension feeding and move by crawling through the water and the sediment. In terms of life history, they are deuterostomes and have a larval stage before they develop into their adult stage.
As I stated before, hemichordates are rarely ever talked about, so how important can they be?
According to the National Institute of Health, studying this group of organisms may benefit human health, as some species within the hemichordate phylum are capable of regenerating anterior and posterior adult body structures after complete amputation. Some of these structures include the heart and kidney complex and stomal cord. Using the knowledge gained from studying the regeneration could give researchers more insight to regenerating damaged or destroyed tissues and organs within humans.
To help us get a better understanding of the evolutionary history of a phylum hemichordata is Chris Cameron. Please welcome Chris Cameron. So, Chris, you brought in some phylogenetic trees. Can you talk us through them, please?
The first tree I brought with me is a 16S phylogenetic tree, which compares a ribosomal subunit that evolves at a slow rate, making it perfect for constructing evolutionary trees. The tree length was 1956, with the outgroup being the Nemertia. In this tree, the closest relative of the Hemichordata was the Chordata. The next tree I constructed was built from a data matrix comparing 9 species. The tree length was 28, and the outgroup was the perch fish, a member of Chordata phylum. In this tree, the closest relative of the Hemichordata is a member of Phylum Annelida.
Interesting. Can you explain some of the differences between these two trees?
So the location of Phylum Hemichordata is different on both trees. This is because having the sequences based on genetics gives a more accurate analysis of its evolution, and because similar structures do not always have the same genetic makeup. Genetic makeup helps us understand the evolution of a taxa better than just physical characteristics.
Well, thank you for your time, Chris. Now we'll go back to Jamie to discuss the scientific paper her and Chris wrote. Let's start off with an easy question. How long has a phylum been around?
According to the fossil records we have, the taxa Hemichordata have existed since the early Cambrian period.
So, Jamie, your paper has a phylogenetic tree in it. Can you explain the hypothesis you used to create this tree?
The first hypothesis is that the chordate ancestor was a moving ciliated larva of an immobile pterobranch-like adult. Gill slits, a notochord, and sexual maturity were transferred to the larva by neoteny, forming a moving protochordate adult that developed into a chordate. Others eventually evolved a collar, stomal cord, and became hemichordata.
Sounds like a lot of research. Is there any part of it that you are still unsure of?
Well, yes, there is uncertainty about the exact evolution of this taxa since more research needs to be done. The evolution from Breckenridge T expression is uncertain since it is unknown whether shared traits are a result of common ancestor or convergence. More research needs to be placed forth about the evolution of this trait, in addition to the development of stomal cord, dorsal hollow nerve, and the pharynx of and Terra Nuest worms.
Between the trees that Chris brought and the trees that you brought in, there are a lot of differences, specifically with the closest relatives. Why do you think that is?
First, I want to make clear that these are both predictions. We don't know anything for sure. However, I had much more genetic code to work with to come up with my phylogenetic tree.
Well, I think we're just about out of time for today. Let's thank Chris and Jamie for coming in and chatting with us. And see you next week on Channel 11 News.